Hello, everyone. Hope everyone's really doing well, and wherever you are in the world and whatever time zone you might be in. First, I'd like to welcome you to our first JCIMinar. This represents really a team effort from the whole JCIM editorial team to put these together in collaboration with ACS Publications, so we're really thankful to them. And also, we're planning on several more of these in the coming months, so stay tuned for the upcoming topics. Before I launch into, Rommie launches into her talk, I'd like to just highlight a couple things that are going on in JCM. First, we have, for the first time ever, we have two special issues in one issue, one on generative methods and one on COVID-19, run by Rommie and Adrian Mulholland. So look out for those. I think they'll be very interesting. We're also changing our policy on data sharing and computer software usage. And we're going to start requiring a data software availability section in your papers. And we just published an editorial on that. It was tweeted. So please take a look at that and make sure you include this section in your upcoming papers. Finally, the last thing that's kind of interesting news is Professor Guowei Wei from Michigan State University has joined us as an associate editor. He's a world expert in machine learning and artificial intelligence is applied to computational chemistry and biology. So we're very excited to have Guowei join us. So it's a real singular pleasure to introduce Rommie Amaro, who's a professor at the Department of Chemistry and Biochemistry at UC San Diego, where she holds a distinguished professorship in theoretical and computational chemistry. Rommie received her BS in chemical engineering and then saw the light, I guess, and moved into getting her PhD in computational chemistry in 2005 from UIUC, where she got her PhD advisor was Zan Luther Schulten. Upon completion of her PhD, she joined Andy McCammon's group as a NIH postdoctoral fellow from 2005 to 2009. And in 2009, she started her independent career. Rommie's been very well decorated over the years. She has a number of awards, including the NIH New Innovator Award, the Presidential Early Career Award for Scientists and Engineers, the ACS Comp Division OpenEye Outstanding Junior Faculty Award, the ACS Kavli Foundation Emerging Leader Lectureship at the ACS meeting a few years ago in San Diego, as well as the Corin Hanch Award. And also, we're very blessed to have her as an associate editor of JCIM as well. Her interests lie at the intersection between computer-aided drug discovery and biophysical simulations. Today's talk will cover kind of that range of things, and it's entitled Molecular Simulations in the Time of COVID-19 and Beyond. And Rommie, before I let you take over, just a couple comments. We won't be able to take questions during the lecture. So if you could please put your questions in the chat. When Rommie finishes, I will then extract the questions from the chat and pose them to Rommie for her answers, okay? So with that, welcome Rommie, and I very much look forward to your talk.
Thanks, Kenny. So good morning or afternoon or evening, depending on where you're joining us. My name is Rommie. As Kenny said, I'm going to tell you today all about molecular simulation in the time of COVID-19 and beyond. I'll try to talk not only about some work that we're doing, but also some other efforts in the community that I think have been really fantastic. Okay, so for those who maybe haven't heard me speak before, my group has been really keenly interested in using a tool called a computational tool called molecular dynamic simulations as a computational microscope. And the idea is that we can combine multiple different types of data sources, from various biological and chemical instruments, for example, structurally, cryo-EM, X-ray crystallography, or tomography data, together with glycomics, lipidomics, and genomics, in order to build highly detailed, biologically accurate physical models of biological systems. And we then, of course, as probably most folks in the audience know, in this crowd, I would guess most folks are familiar, but we basically are approximating that system down to its many atoms. And we define a potential function that I'm showing you here that sort of defines the interactions of the different atoms in our system. And then all that we're doing is essentially integrating Newton's equation of motion over time to explore the time-dependent classical dynamics of our system. And we run this integration, this numerical integration on typically the work that I'll tell you mainly about today really relied heavily on the supercomputing resources in the US, two machines in particular, the TAC Frontera machine, as well as the Oak Ridge Summit machine, were really key to some of the things that I'm going to tell you about. But I think what I hope shines through in the talk is really, how nowadays we can use these simulations as a data-centric way to explore biological systems. And what I find really fascinating is how we can really use them to predictively and prospectively get ahead of where experiments can go. So, before January or February, my group had been working for a number of years with the influenza glycoproteins. And one work in particular sort of came to fruition in a key time of this story that I'm telling. And this was sort of in mid-February or so. We published finally, after a number of years, it took us about 7 years, a study describing an all-atom simulation of the influenza viral envelope. This work was published in ACS Central Science. We were very excited. It got one of the covers. And that same week that it was published, you can see that what I'm showing here are the total confirmed COVID-19 cases in the US as a function of date. So starting back in January in different countries. In mid-February, the United States was still pretty low in terms of the number of cases, had about a dozen cases. And even though it didn't, look particularly bad in the US, we knew at that time that the virus had already escaped mainland China, and that actually that the Lombardy region of Italy was getting really hit very hard by the virus. So this is when we started, I started really sort of thinking maybe we need to do something with this with this other type of virus. And in fact, it was, again, just that same week that the group of Jason McClellan at the University of Texas, Austin, deposited a preprint into the bioarchive, which described the first cryo-electron microscopy structure of the SARS-CoV-2 spike protein. And this was the day, when this was deposited, this was really the day that sort of everything changed for my group. And I just also want to sort of mention how these preprint servers, particularly in the space of COVID-19, have been really critical to advancing research in this space as rapidly as they have. And I think, just sort of hot on the heels of having two vaccines being approved in the United States, it's really incredible to think that just, not even a year ago, just about a year ago, we were first hearing of this virus. And now to have what looked like at least two, probably more really effective vaccines is just incredible, frankly. And a lot of that has been, or all of it has, a lot of that has been due to this sort of, I think, really fantastic sort of science efforts. So in any case, this was when we sort of really started to turn our attention to SARS-CoV-2 that same week. And then, I have about 40 minutes, so I can't really tell sort of like all the things that we've been working on, but I wanted just to give a timeline of some of the key events in our studies. And so again, in early or mid-February was when the first cryo-EM images of the SARS-CoV-2 spike protein were released. And then from there, you can see some of the different types of things that we've worked on. Our first tweet, so it was sort of interesting for me because I also joined Twitter right around when, right before the ACS Central Science publication for influenza came. So that's been another sort of method for scientists to sort of stay abreast of all the advances. But from this first tweet, which was in mid-March or so, through October, we've been able to sort of carry out a number of different simulations and different methods together with a whole bunch of collaborators, which I'll talk a little bit about some of these different areas that we've worked on. But it's really been, it's been an incredible run for the past, how long has it been? I guess 9-10 months in the group. So most people, I'm sure here are already familiar with the SARS-CoV-2 virus. I'm showing you here an all-atom reconstruction of that virus. And what I just want to point your attention to is, one of our primary sort of areas of concern, of course, is the so-called spike protein, which you're seeing here sort of sticking out of the surface of the virus. And, the spike protein is so important because it's the first point of contact that the virus makes with the host cell. And so, and it also, because it sits on the outside of the virus, it's really one of the main sort of antigenic molecules that the virus has. And, many of the vaccine constructs actually utilize the spike as its sort of main component. And so we and others, there's many people interested in understanding really how the spike protein works and sort of trying to unlock its secrets in terms of understanding viral mechanisms of infection. And so the other really important part of the spike is that in order to infect the cell, it needs to make contact with a molecule on the host cell called ACE2. And there may, in fact, be other receptors that the spike protein interacts with. There are several studies that point to some additional receptors that may potentially be recognized. But ACE2 really seems to clearly be the predominant sort of mechanism of entry. And so what I'm showing you here is the spike protein here on the viral membrane. And then up top here is ACE2 coming off the host cell membrane in yellow. And what I'm going to come back to a number of times is this light blue bit in the middle here. This blue bit is something called the receptor binding domain. And this is the part of the spike protein that makes this critical interaction with ACE2. I like to think of this as a handshake between two people. This is the interaction that really must form in order for the spike to actually infect the host cell. And so we'll keep coming back to this receptor binding domain and this interaction throughout the talk. So, really from the very beginning, when we first learned about the virus, sort of again, coming sort of fresh off the heels of the influenza work, we thought it would be fun, in fact, yes, and very interesting to try to repeat what we had done with influenza, but now for SARS-CoV-2. And so in order to do that, we start with, one doesn't just build the virus, the whole virion, sort of all at once. The strategy is, at least for us, has been that one sort of deconstructs the virus into its molecular piece parts, build high quality individual models of each of those, make sure that each of those models is sort of, experimentally validated and high quality, and then bring those pieces together into the working whole. So for us, that meant starting, of course, with the spike protein. And so most of what I'm going to tell you about today involves the spike protein and what we've learned. So the spike protein is a very large molecular machine. It is a trimer protein. It has 3 monomers that have about 1,300 residues each. You can see here some different important domains that we talk about: the N-terminal domain, the receptor binding domain, or the RBD. I don't have time to go into all of the many details of this, but just to give you a sense for the architecture, there's a large head region which takes up almost, a little over 1,100 residues, forms the head of the spike. And then there's another region, which I'll tell you about, the stalk and the cytoplasmic tail, that basically connect that head to the viral membrane. So again, the first structures of the spike head came out formally in the journals in March, in mid-March and late March or April. The first study by the group of Jason McClellan, where they determined with just such impressive speed a very nice structure of the head and one of the key things that they found was that one of these receptor binding domains actually was in the open conformation, and you can see this green bit sort of sticking up. A few weeks later, the group of David Wiesler at the University of Washington also put forth a beautiful study where they also recapitulated the open structure of the spike, but they also used symmetry to develop a model of the closed spike. And so, these early studies actually were missing a number of loops and so forth because they were too dynamic, essentially. We built those back in using sort of standard sort of homology modeling approaches using Modeler. And we're left at the end of the day with two, I think, very nice models of the head. As you can see here, one with the RBD in the up conformation and another in the open conformation and also the closed conformation. So, but as I mentioned, we wanted to really build the complete spike protein. So, we had the structural information from cryo-EM for the head, but we also then wanted to join this together with models of the stalk. The stalk is essentially known to be a trimeric coil-coil helical bundle. We created that with Modeler, where it's basically just a series of helices with some bendable joints. And then the cytoplasmic tail, we also modeled, we modeled that using iTassar. And then we brought all of these, different units together to build the complete spike protein. And here I've sort of tried to color code the sequence domains up top, also, structurally, so you can sort of see where they map. But another sort of really interesting feature and a place where computing and simulations has a lot to give is in regards to these, amazing post-translational modifications known as glycans. And so, as beautiful as those structures were with the cryo-EM of the spike protein, the spike protein is actually covered in a sort of series of post-translational modifications known as glycans. This is basically a sugary coating that essentially masks the spike from the human immune system. And this is really what allows the virus to go undetected in the human body and to be essentially so effective in terms of infecting. And so, but the issue is that they can't actually, structural biologists can't see or resolve the structure of the glycans. And this is in part because they're basically too flexible. So the glycans are there, but they can only typically resolve the first unit of the sugar. And so we use simulations to essentially combine glycomics data. In particular, we use two different really excellent studies, one from Max Crispin and Jason McClelland that was published in Science. I guess this was maybe in March or April. And then also we used the data from Parastou Azadi's group from the University of Georgia, who also presented some O-linked glycans. And we basically then, so these studies give us at each of the different sites on the of the spike protein, it gives us sort of the molecular composition of those glycans. And so we can then rebuild them using atomic level modeling. And so I just want to give you a sense for what that looks like. So, there are these predominantly, there's 22 N-linked glycans. And so you can see the different asparagines here that are known to be glycosylated. They, again, from the glycomics data, can determine or inform on what type of glycans those are. And then we can actually rebuild those glycans bit by bit, piece by piece into these models. So you have not only the 1,300 residues for each of the monomers, so 3,000 over, almost 4,000 residues of protein, but then you also have these large branch structures, of these sugars. And I have to say, this is in chain A and chain B and chain C. This is really, really very painstaking work, but important because you can see that there are a lot of them. And so, it's simply not an option. If one wants to make accurate models of the spike, it's simply not an option to ignore them or to pretend that they essentially don't exist. They really need to be included. So they were in there, our models included such details. As I mentioned, we also, in order to build a full model, we also needed to come up with something for the membrane. And so the virus actually buds off of the endoplasmic reticulum and in a compartment called the Golgi intermediate compartment. And so, thanks to many years of biology, we sort of know the relative membrane composition in terms of the lipids and other molecules that compose these membranes. And so our models follow those molecular recipes relatively closely. You can see here the different lipids that we've included, as well as cholesterol in our models. And so here now I'm showing a sort of the slab of the membrane, and you can see the different lipids colored in pink and fuchsia and orange and yellow. So you can see how they're all intermixed. And then the last thing I want to draw your attention to is this area here. So this is the transmembrane region. And that cytoplasmic tail that I mentioned before, it's actually, there's so many interesting bits to this protein. It's just, it's really amazing. But that cytoplasmic tail has, it's littered with cysteine residues. And those cysteine residues are post-translationally modified as shown here. So there's palmitylation groups that post-translational modifications that essentially these sort of fatty groups, they basically sort of snorkel up into the membrane and help anchor the spike protein, into the membrane. So our model has those also. And then, so this is what the reconstruction looks like at the end of the day, just taking you on a quick tour through that. We have all of the domains, as I told you about. We also have multiple structures. We have, in this case, I'm showing the open structure or the up structure, but we also have the closed structure from the Wiesler group. And then you can see all of the glycans, and again, sort of decorating the entire end-to-end structure. We have the membrane, the palmitoylated cysteines, and then this is what the fully reconstructed model looks like at the end of the day. And then we essentially are animating or predicting the atomic level dynamics, so the wiggling and jiggling of the atoms in our system using all atom molecular dynamics. And so the wiggling and jiggling that you're seeing here are those structures from the molecular dynamics simulations. And you can really see how the glycans, which at this point almost look like ornaments on a Christmas tree, how they're sort of decorating the whole structure. And so these systems, I'll just briefly tell you, they have about 1.7 million atoms, and we were using the Charm 36 force field with NAMD2 on the TAC Frontera machine, and on 256 nodes, we were able to get about 60 nanoseconds a day. And we gathered over 4 microseconds of data for each system, and I'll tell you a little bit about some different systems. So what did we learn from this? So the main thing that I think was really one of the major findings of our study was that we showed the world for the first time what the full glycan shield actually looked like. And so on the left in just blue is sort of the naked protein, or the protein without any sugars. And so this is the view that essentially that cryo-electron microscopy gives us. It's just sort of what the protein itself looks like. And on the right, what I'm showing is a picture of the spike, but now also shown in dark blue are these glycans. And you can really see, I mean, it's so striking. You can see how different the spike protein looks if you're just thinking about the protein and if you actually are having the glycans there. And so this was really amazing for people to see how shielded it is. So they we call these glycans a glycan shield because that's what they're doing. They're basically masking this foreign agent from the human immune system by covering itself in these sugars that are also somewhat similar to what's found in other proteins and on host cells. And so people are very interested not only to know what the shield looks like, but of course we're also very interested to know where the shield isn't. Right, because these holes in the shield represent vulnerabilities in terms of the ability for us to develop neutralizing antibodies or approaches to vaccines, as well as potentially novel drugs or therapeutics, that it's easier to access these areas that aren't covered in glycans. So our simulations also showed why the spike actually opens and closes. And so what I'm showing here on the left is an image of the closed spike looking from the top down, and in the top panel, and on the bottom, a side view of the top. And so I like to think of the closed spike really as the defending mode. If you look at the open spike, again from the top, you can see really clearly how this receptor binding domain in light blue, this was that part of the spike protein that must make that critical interaction with ACE2. You can see how it's really now it's in an exposed state. And again, here from the side, it's really dramatic I think from the side, comparing the closed spike structure the open spike structure, how this is really the attacking mode or the exposed mode of the spike, whereas in the closed state, it's really in the defending mode, and that it's shielded or down. And so I also found that really just fascinating, because otherwise, if you're just looking at the structures from cryo-EM, of course you call it up and down because that's what you see. But with the glycans there, I think a whole better nomenclature would have been exposed and shielded, or defending and attack, in terms of the modes of the sort of function, relating structure more to actual function of the spike. But so, of course, these data sets are huge, and they have, these time points every step along the way for microseconds of dynamics. And so, it's really a treasure trove of data, which we've shared widely. I'll talk about that later. But so one of the key things that we and others are interested in is something called the solvent accessible surface area, or the accessible surface area of the spike, which I'm showing here. I'm plotting in terms of the square angstrom as a function of probe radius, where each of these points represents, over a microsecond of dynamics, sort of what is the average exposure of the spike protein, or in other words, how much protection do the glycans actually sort of impart on the spike protein for probes of different sizes. Because of course, if it's a water molecule or a small molecule, these will be more effective at gaining access to the spike than something that's much larger. For example, this would be up here on the right end, between 14 and 16 angstroms would be sort of the relative probe radius of an antibody, a neutralizing antibody, and you can see how much more effectively shielded the spike is. We did that not only for the head, but we also looked at the stalk. And so you can see just by looking at it that this stalk portion, which they can't resolve in cryo-EM, is really shielded. And upwards here, you can see upwards of 90% of that stalk is shielded from access by antibodies. And the stalk is really an interesting area. I mean, a lot of folks are interested in this because it's one of the most conserved regions of the beta coronaviruses. So it represents a potential place where broadly neutralizing vaccines could potentially be sort of developed to target that area and sort of make these pan coronavirus type therapeutics. But it's the fact that it's shielded, it's good to know. It could suggest that it would be sort of a difficult strategy to undergo. So the other thing that we've done is sort of study, we're able to sort of study now where these different neutralizing antibodies are binding. And so what I'm showing here in these different colors with these labels, so like in orange is the 47D11 antibody. This targets the receptor binding domain, same as in red, this B38 neutralizing antibody. And we've just sort of gone through for a series of some of the earlier structurally resolved neutralizing antibodies. We've mapped onto the surface of the spike where those neutralizing antibodies are binding. And you can see that, yes, there's a huge number that are in fact binding the receptor binding domain. And thus they're probably working by sterically hindering that interaction with ACE2 on the host cell. But there are some other places where neutralizing antibodies are binding. For example, out here, this is an N-terminal domain neutralizing antibody called 4A8. It also, you'll note, interestingly, most of the antibodies are binding directly to the protein. This one's actually a glycan, is recognizing a glycan or one or two glycans there. So that's interesting. And then also there are some that, for example, there's one antibody, 1A9, that actually is binding down in the near the connecting domain, which is the part of it's sort of like far down on the spike head near where it's meeting the stalk. And so it seems apparent that when one looks at this, the epitope where this is known to bind, it's quite buried in this so-called pre-fusion state. So it suggests that these antibodies are recognizing a different state along that the spike adopts, conformational state, right? And so we're focused in this work very much on what we call the pre-fusion state of the spike, which is sort of what it looks like as it's sitting on the viral envelope. But of course, this this protein, it's really helping to mediate viral fusion with the host cell membrane. And as it goes through that process, undergoes an enormous set of conformational changes. So in some sense, our studies are quite limited, in fact, in terms of just being able to really investigate these initial states of the spike protein and in the infection process. So, the shield is very interesting, but something even more fascinating sort of jumped out at us as we were studying this protein. And I want to also just mention this is a collaboration. This work has been a collaboration with Elisa Vette at Maynooth University. Excuse me. She's a fantastic glycobiologist, glycochemist at Maynooth University. And when we were building this structure, something that jumped out at us was that when we were looking at the closed spike structure, there was one glycan in particular that this is at position N234, that we always saw was sort of sticking out into solution. And so looking down at the spike head, you can see how it's sort of sticking outwards. And then when we looked at the open state, we actually saw that one of these glycans was actually oriented inwards. So we looked a little closer and we realized that in the open state of the spike, there were two glycans that, excuse me, there were two glycans that sort of went down underneath this, the RBD, and basically propped it up. Excuse me, I have to take a drink. I apologize. So we came with the idea that what could it be possible to basically mutate these two residues? And we did that. We mutated these residues to alanine. Oh my gosh, I'm losing my voice. And we ran multiple microseconds of dynamics for that system. And our hypothesis was that if we mutated those residues, that RBD would basically collapse down and it would reduce the interaction with ACE2. And so, in fact, that's what we saw with the dynamics. We actually identified, so there's two different sort of motions that we did a principal component analysis, and there were two different motions that we were able to sort of discern. One is something we call the axial tilting, which is if you're looking at the side, sort of how the RBD actually sort of folds in and out or up and down. And so we saw that in the closed state, nothing really happens. In the open wild type state, it's overall fairly stable. But when we introduced those mutations, it actually sort of closed down a bit and sort of, it did fold down on itself. Another major motion that was identified with principal component analysis was something that we defined as the lateral tilting. And so this was actually sort of an in-plane motion of the RBD, sort of a twist. And again, when you look at the closed state, nothing much happens. The closed wild type, if you look at the open wild type, again, it's fairly stable. But the mutant structures actually were highly unstabilized and adopted, in fact, sort of a bimodal distribution. So there was a much greater range of motion. So then, we just did something not crazy, but we basically called up, we felt like this was a pretty solid hypothesis with some data to back it up. So I contacted Jason McClellan, who I'd never spoken with before at the University of Texas. He was the cryo-electron microscopist who came with the first structure of the spike. And I just asked him, I told him what we found. And I asked him if they would be up for doing some experiments. And he said, yeah, absolutely. That sounds cool. So they developed a biolayer interferometry experiment to test the behavior of these mutants. They have, and basically we can watch how or monitor how the spike protein interacts with ACE2 over time. And they have this for, there's two controls. One, this cysteine mutated version of the spike basically is like a lockdown conformation, so it never interacts with ACE2. The wild type here is shown in green, and then two of the mutants, they were individual mutants that they made, single point mutants. You can see that's the reduction in binding. So that was fantastic. So, this work not only showed the shielding of the spike, but even more I think strikingly showed for the first time it established a structural mechanical role for glycans in the system. Actually, I think it's the case it's the first time that a mechanical role of glycans in the function has ever been shown for a protein, which is really, really cool. And so we showed that these glycans not only shield the spike protein, but they also act as part of the viral weaponry itself, essentially locking and loading the spike for infection. This work was published in mid to late October. Since then, we have, we sort of continued to study the other parts of the viral interaction with the host cell. So I'm showing here a simulation of ACE2 in a cell membrane. This work was published recently in, excuse me, Biophysical Journal. One of the things that we see is this really extreme degree of spontaneous tilting of ACE2 relative to the membrane. And we sort of speculate that this high degree of tilting helps to sort of accommodate the sort of large mechanical motions that the spike has to undergo during the cell fusion process with the virus. And then, the other thing that we're interested to do, so everything else that I'm showing you now is unpublished work, although it was disclosed in our Gordon Bell preprint. But something else that we're interested to do really is understand that transition from the fully closed to the open state. And, unfortunately, it would take years to do this with standard molecular dynamic simulations because it's a very sort of slow process. So we joined together with Lillian Chong and her team at the University of Pittsburgh to explore other ways of being able to sort of help open the spike structure. And so she has this because in molecular dynamics simulations, we essentially spend a lot of our time in these energetic wells and really not enough time sort of going over barriers and so forth. So we spend a lot of time sort of waiting for things to happen. So she has this method called this weighted ensemble molecular dynamics method, which allows researchers to sort of really focus on functional transitions. And essentially what it does is sort of like separates something that you define as a progress coordinate into different bins, and then sort of restarts trajectories as they cross into these different bins. And so at the end of the day, what it does is it allows you to generate continuous, unbiased pathways, in this case, unbiased opening pathways, of that spike closed to open transition with orders of magnitude less computing than standard MD. And the thing that's really one of the real advantages of this method is that the higher the that barrier gets, the more efficient her method becomes. So it's really a really powerful way to gain insight into these sort of pathways and to capture transient states that are really too fleeting for experimental characterization. So we used her method, which has been implemented in a tool called WESPA. And this is something that's essentially embarrassingly parallel. So for this, instead of sort of, putting out, one simulation to 256 nodes like we did with Frontera, for this, what we did was we launched many thousands of short timescale runs on hundreds of GPUs. So in this case, we were using the TAC Longhorn partition, which has hundreds or thousands of NVIDIA V100 cards, and we were able to generate about 133, so hundreds essentially of independent opening pathways in about 23 days of compute wall clock time versus, if we wanted to, for example, achieve that same number of pathways with standard MD, it would have taken us potentially, it could take us at least a year. It probably would have taken multiple years and taken a lot more node hours. So it has a real sense of efficiency to it. And then I'll just sort of briefly mention as we continue. So now that we have this so the weighted ensemble method really is showing us in full atomic detail what these transition pathways look like. And of course, we are exploring different mutations to test some of the findings from these simulations. But then, once it's open, we're also really interested, of course, to study, how it's interacting with ACE2. So we have also generated now, again, this part is unpublished work, structures of or simulations of the interaction of the spike protein with ACE2 in a two membrane system. And so we can really sort of get insights into what's happening at that interface. And also, we're looking very much at what happens with this sort of stalk bending, which Gerhard Humer also saw in some of his simulations at the larger scale also. And then something that we've sort of recently achieved, as I mentioned, of course, we're very interested to get to the level of simulating the whole virus. And really to do that, the single biggest obstacle to simulating things like viruses is getting the membrane right. And, that's really because differences in the inner and outer leaflets that are induced by the curvature sort of like make all of these sort of interesting blips happen, holes form and so forth. So what's required is really an intensive iterative remodeling process where one has to, generate an initial model, run some dynamics, fill in the holes, et cetera, do this sort of patching algorithms. And so we were able to do this. In fact, Abigail Dahmer, who's in my group, this is really a heroic effort, because I would say this same sequence of events that for the flu took us, probably, on the order of years, 3 or 4 years to actually do correctly, she was able to really much more rapidly achieve that for SARS-CoV-2. So now we have this up and running. It took I guess about 7 months for her to achieve that, which I think is really, really heroic. And we've been able to run that simulation. So this is a 305 million atom simulation. We've been able to get that up and running on the Summit, on both Frontera and the Oak Ridge Summit supercomputer, where we see, strong scaling with NAMD. And so we're just now getting into analyzing those trajectories, which of course will be really rich with data. And again, at the virion model, at the virion scale, the patterning and the density of those spike proteins is taken directly from the tomography experiments. There was a beautiful series of 4 experiments characterizing the virus in near molecular detail from groups all across the world. And we're using that data to we've used that data to construct these sort of faithful representations in silico. So, with that, I wanted to sort of pivot in the last, because I do want to leave time for questions. The last few minutes, I just want to say what a special time this has been for molecular simulation and for scientists in general. And, we recognized early on the importance of being able to communicate openly and with other scientists working in this space. And so, Adrian Mulholland, who's a terrific colleague at the University of Bristol, we just started talking about it, and we realized that there was really a need to develop maybe a set to do things differently for COVID-19. And so we worked together with many people in the community to write a letter, which was published in JCIM in March, I think, in late March, where we outlined a set of principles that we would commit to, which would include making our work on SARS-CoV-2 available as preprints as soon as they're submitted to journal, but make them available to everyone, to deposit models at the time of deposition into the bioarchive, make models available then, which again is really remarkable, and committing to making available input files, processing scripts, sort of really everything that's needed in order to really study these systems. And I was just really sort of it's really just remarkable, sort of the warm reception that this received. Over 200 different groups all around the world signed on in committing to these principles. It was really embraced by the community, which was fantastic. Another really key thing that happened was, something that we don't often talk about, but that was really crucial to scientists being able to move very quickly into the space, was the availability of really high quality open access community software. such that, as soon as this threat hit, we were able to use a number of different softwares in order to, to propel forward rapidly sort of what we could learn about this system. And so for us, we really primarily used NAMD and VMD, which were fantastic tools. But there's many different, many other groups have used other community-based tools, and I just want to sort of emphasize on the importance of those efforts, where it's really been for many of these sort of decades worth of efforts to make these codes available. The other thing that was, of course, really special and that really was important for our work was the COVID-19 High Performance Computing Consortium. Right. And so this was something that came together very, very rapidly early on. Again, this was in March. It came together early on. So, where the computing sites recognized that they needed to get resources to the community and to do that very quickly. And so they, banded together and created this consortium that really rapidly reduced the amount of time required to actually gain access to such resources. And I would say this also was really, really key. I mean, for us, I don't think I mentioned this in the beginning, but that day that JSON structure became available in February, I walked down to Lorenzo and Ziad's office and I said, okay, guys, we need to pick it up now. And then when I walked back to my office, I immediately contacted Dan Stanzione, who is the director of TAC. And I said, Dan, there's this virus. And of course, at that point, we had already heard quite a bit about it in the news, and even we knew what was happening in Italy. And I said, I think we need to do something about it. Can I get access to Frontera to try to repeat what we just did with the flu? And he said, absolutely. He was very gracious and just generous and said, absolutely, Rommie, you can do this. Whatever you need. We just let me, help you. And that was really, since then, we've had essentially unfettered access to Frontera and to many of these different resources across the US. And also this has been something not only in the US, but in, across the world where the supercomputing sites have become available. And that's been fantastic for efforts in this space. Whereas otherwise we would need to wait probably upwards, almost, at least 6 to 8 months before we could actually get access to these machines because of the review cycles. We could get access instead in about 7 days. The other thing that was really key for us, and I hope for others, was that in the spirit of really sort of collaborating and working together, one of the things that we did was, again, share our model very early on. And so, something that I tend to be a little bit critical of our field, we don't share as much as we should, as much as we know we should. And I mean, sharing is important for things like reproducibility. And in the case of COVID, it was really important just to make sure that the models that we were developing were as good as they can be. So to have, so in this case, we made our models available way early. So in March, we started sharing our fully glycosylated spike protein. And I'm grateful to all of these groups. I didn't write their names, but these are all fantastic colleagues from all over the world, top folks who took the time with their groups to look at the structures, to check whether or not, we had all of our correct disulfide bridges, the protonation states on our histidines, to make sure that the glycans were connected properly. I mean, when you have 1.7 million atoms and you're trying to do things very rapidly, there's a lot of places where errors can come in. And we certainly are very grateful to the scientists all around the world who helped us to, improve on our model and to spend time to really look at it. And that was really, it's been a special experience, I feel like working in this space. And then the other thing that the community letter we published in JCIM helped to formulate and sort of helped to inspire the development of is this COVID-19 molecular structure and therapeutics data sharing hub, which has been developed by MolSI as well as BioExcel in Europe and other organizations. So there's now sort of a central repository for the first time, a central repository for molecular simulation data, as well as other data sets, involving the SARS-CoV-2 virus. And so our data in simulation, it's big. So the weighted ensemble trajectories that we have is about 200 terabytes of data. The whole virion simulation, just the PDB structure of the virus, even without waters, is multiple gigabytes. So it's it's difficult to share our data, but we as a field, we really need to figure out ways to do that. And I'm sort of really grateful and happy to see when push comes to shove, that the ability of our community to actually develop such sharing sites, it was done quickly, and now serves as a place where the exchange of data can be faithfully carried out. And so the Frontera data sets that we've developed have been shared over 4,000 times with members of the community. I know it's been accessed by people at various pharmaceutical companies, people working on vaccine development, and others, who are now harvesting in that data. And I think that's good. It just sort of amplifies the reuse and helps us again sort of move forward on the science better. And I think this is one of the things that we really hope sort of sticks. It's one of the things that I hope sort of sticks with the field, is sort of beyond COVID-19, this notion of sharing our data, not just our data, but our input models and so forth. And to do that, and the adoption of the use of preprints, I think these have all been really good things that I hope stay with us as we move forward. And then I just want to mention at the end, before acknowledgements, I am an associate editor at JCIM. It's a fantastic journal, that is really rising in the ranks, which is terrific to see. And together with guest editor Adrian Mulholland at the University of Bristol, I guess inspired in part by the fact that we helped to write that community letter, we at the same time invited contributions for a special issue, which I'm showing here, which will be published together with another special issue on generative models for molecular design. But there's a number of really top contributions from colleagues across the world in using computational methods to study the virus. So please check that out. And with that, I've made it through my coughing fit, I'm happy to say. I wasn't sure if I was going to make it, but I did want to leave time for questions. So we have about 10 minutes. I just want to thank everybody. I mean, it's been such an incredible 9 or 10 months. Really just sort of one of those unforgettable things that you go through. I want to especially thank Lorenzo Castellino, Ziad Gayeb, and Abigail Dahmer, and Tara Stain, and also Shirley An. These have been folks in my group who have just worked absolutely tirelessly. Also, Amelia, where is she? She must be in here somewhere. Yeah, there she is. Amelia, who's now gone on to a postdoc. They've just so many sleepless nights. I'm so proud of this whole team. And then also Elisa Feta at Maynooth University, terrific glycobiologist. I mean, these glycans are really something to be reckoned with. As you can see, they are quite complex. She's just fantastic. And many others all across the world for their help again. And I thank you for your attention, and I will be really happy to take any questions you have or to have other discussions.
Okay, well, thank you very much, Rommie. That was a really stimulating talk. I don't know if I really want my face associated with an infection point of a virus, but that's really cute.
That's a good point.
Yeah, but so anyway, so there's a couple questions. I think one was the availability of your lecture. I believe that's going to be made available maybe on ACS Axial. I don't know if Jay or somebody wants to chime in. So that was one question. So the first question I'll pose to you, this is from David Ramirez, and I think you sort of answered this, but anyway, is it possible to access the PDB files of both the spike and spike ACE2 complex for further docking simulations and virtual screening campaigns? It is by far the best plebe mod I've seen so far. Wonderful work.
Oh, thanks. Thank you. So yes, on the spike. So the spike protein we have made available through that multi site, and also you can get to it from our homepage, and it links also at the journal. We have those. The ACE2 complex we have not released yet. And that was really just because, so we will release that. We will release that, but we're still in the stage of gathering the data. So we're not finished actually running all of those trajectories, the ones with the parallel two membrane system. The weighted ensemble trajectories, I think there's going to be a lot of interest for those because we have, I mean, it's amazing, this transition. And of course, one can think about the possibility to develop molecules that sort of freeze the transition into these different conformational states. Those will become available very soon. So I think I'm hoping that we can get this. So what we're doing is as soon as we put the preprint out, then we're making the data available. And the reason for that is really that one thing I'm very sensitive to is putting out data that's not good. You know, I don't want to do that. We don't need any more sort of bad data anywhere, but especially in the space of COVID-19, we've seen some, you know, really interesting things. So there's a certain amount of vetting that we do internally, and then also with, you know, collaborators, as I've showed you. And so we want to make sure the data is to that point. And then also when you have that preprint associated with the data, the methods are fully explained. So you know exactly how we treated the system and, you know, what the strategies were, as opposed to just sort of releasing it without a careful documentation of what it actually represents. So yes, all of it will become available. It's just a matter of time. And, you know, it's sort of, you will see additional data coming forth from this group in particular, I'm sure, like over the coming months.
And this will be all made available at SSI site. Yep. Fantastic. So the next question is from Holly. Have you looked at the VUI-2012/01 strain yet? I assume you know, I don't know what that is, but, or can you make any predictions about N501Y mutant?
Oh, so this is the one that's just circulating in the UK. Okay, so no, we haven't simulated that yet. So, you know, we started with those the initial structures. It was one of the initial Wuhan sequences. So the simulations that I'm showing you also don't have that D614 or 641G mutation, which is the circulating one, I think predominantly in the United States, the circulating strain. So these are all things that can be explored and are being explored gratefully by others. So I can't say anything yet. I mean, the D641G mutation does seem to change some of the dynamics according to some colleagues. There might be, you know, I think there's some interesting things there. 501, we have not yet studied. But I also want to say something else. I mean, I think this is why it is really good for many groups to study this protein, because there are just too many questions for any one group to go after. And so similarly with the glycans, it's like everybody who builds the system, they'll each bring sort of, I think many groups each will bring their own level of expertise, and it vary the model a little bit, whether it's the sequence of the amino acids or the glycans or, you know, protonation states. And so then at the end of the day, we're going to have really sort of a set or an ensemble of models that we can all learn from. And so I think that's also going to be one of the sort of collective benefits from this, a silver lining of this pandemic will be the ability to sort of do a meta learning exercise on everything. And of course, AI also might be something that would be really useful in those type of situations. Anyway.
Yeah, and also force field variations too, right?
Yeah, absolutely.
So Sergio Pantano, thanks for the talk. Did you check if the RBD motion in different protomers is correlated? Could the opening be cooperative?
Very good question about cooperative opening. You know, right now, so this initial study that most of what I told you about was just looking at sort of the low level dynamics around two different conformational states. And you really see in the mutant state, you see the destabilization, but you don't actually see any kind of opening. So I don't have the data really to say whether or not it's correlated. With the weighted ensemble, there also, we were only able to look at the opening of one RBD. And of course, there are lots of interesting unanswered questions about, you know, well, once one RBD opens, does it become easier for the second RBD to open, and so forth. But those are all unexplored as far as I know. So it's too soon to say, but a good question.
Okay. The ubiquitous anonymous attendee asks, for molecular docketing, which form is better to start with, the open or closed form of the spike?
Oh, well, it depends what you want to do.
Yeah.
Depends what you want to do. I mean, yeah, you might want to consider if you're talking about, when you say molecular, if you're talking about small molecule docking, I mean, a lot of folks have the strategy to try to dock to that closed conformation to lock it closed, because if it's closed, it's not going to be able to bind to the host cell. I think that's one strategy. Although others are interested in targeting the open conformation, because it's possible to sort of lock the dynamics such that it can't, undergo the rest of the steps, for fusion. And again, these have all been these are all areas that I know people are going after, but we're still in the throes of figuring out,
Really.
different ways that chemical probes can affect function of this amazing molecular machine.
Yeah, so David Ramirez had a follow-on, but I think it was related to the docking, so I think you addressed it. But certainly feel free to contact Rommie, David, if you have your answer wasn't fully answered. We're going to try to end right at 12, and we're going to let Adrian Mulholland have the last question. Thanks for a real beautiful talk. Can you comment on the potential effects of emerging spike variants, deletion, mutations? Do they affect glycosylation? And are they going to affect the different antibodies? That's sort of my add-on to that.
Yes. So I hear your clock in the background, Kenny. That's great.
Yeah, my grandfather.
Thanks. That's nice. Yeah, thanks, Adrian, for that question. I mean, certainly one of the things that we're sort of always on the lookout for is whether or not mutations are, in fact, introducing, for example, a new glycan sequence, or, you can have, I think there is evidence in patients of having even larger scale type mutations, for example, large deletions or insertions in loops and so forth. Yeah, I mean, that's what can I say about it? I mean, I think, there's a lot of interesting things to study about this spike protein. We're going to see more and more mutations, of course, as we go forward in time. And certainly there will be some that are of importance for, with various therapeutic implications. And so I think it's going to be just a matter of, time before we get our hands on enough data that we can explore such things in real detail.
Okay, well, Rommie, thank you so much. It was a really wonderful and timely talk. And thank you everyone for attending. Yes, the talk will be made available. I assume that will be, some link will be made available for everybody. And again, thank you so much, Rommie. Really appreciated your time.
Yeah, thanks. That was fun.
Yep. Okay, see you everybody. Stay safe. Avoid spiky balls, unlike Rommie. They seem to be everywhere for you. So
they are indeed. Take care, everyone.
See you, everybody.